shop owner has bought in the Santa or in any Raitu Bazaar.
एक ही जगह पे मिलके those type of plastics are known as thermosetting plastics. Comparison, there are 3 types:
up, down, right, left.
I am not inferior to anyone.
We are Taurus. We are top on the Everest. Congratulations.
I want to be a leader.
Good morning everybody. I am TSWREIS studying in Kammadanam at Mahbubnagar district. I am going to teach us topic from botany. First I would like to ask you some questions. What is the main occupation of our country? Yes, you are right. Agriculture is the main occupation of our country. So much of energies are required for 62, in our country 62% of population are employees of agriculture. But the food product is only 33% of food production. So much of energies are required for energies and economy are involved in agriculture. But are we satisfied with the food products obtained? No. Why? Population explosion is the clear-cut reason. Though many ways of population controlling methods are used, but population is increasing day by day. And we are forced to invent various strategies for enhancement of food production. All of us know that according to Malthus, he was an economist. Population is increasing in a geometric way, but the food production is in an arithmetic way. For example, food production. Here the numbers are is 2 becomes 4, 4 becomes 6, 6 becomes 8, so on. And the population is, here the population order is 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32. That means here food production number order is in an adding sequence, but the population sequence is in an multiplying sequence. So, the food produced are not sufficient for growing populations, so we want to maintain various different cultures of methods other than our traditional methods. For example, that is why our scientists like M.S. Swaminathan in 19th century taught himself various strategies for enhancement of food production, like plant breeding techniques, mass selection, hybridization techniques, etc. in the name of green revolution to produce high yielding varieties. We also know that food production is the population of, we are all know that population is increasing day by day. That is why whichever strategies are followed earlier are not sufficient for fulfilling our needs. That is why there is a necessity of the people to enter into the cytological problems. What are the cytological techniques? Yes, the cytological techniques are electron microscope, X-ray diffraction, etc. After traditional breeding techniques, we are inadequate to keep with the pace of demand of food production. We are able to produce a many number of food productions. Suppose using technologies like cells, cell constituents at the industrial level of the essential for the useful products and essential to human life. And we are going to teach, now our topic is tissue culture. There are several new techniques like mutation breeding, tissue culture, rDNA techniques, etc. These are playing pivotal role further enhancing the food production. Now, our topic is tissue culture. Now, let us go deep into the tissue culture. Before going to this, let us know about what is a tissue culture. Tissue culture means the whole plants could be regenerated from any part of a plant. It is called in an under sterile condition in a test tube. It is called tissue culture. Here the whole plants could be regenerated from any part of a plant and taken out in a test tube under sterile conditions in a specified nutrient medium. This is about tissue culture. In tissue culture, there are different types of tissue cultures: embryo culture, cell culture, seed culture, organ culture, protoplast culture, meristem culture and seed culture. These are the parts of the tissue culture, and this can be used for culturing, and it is completely laboratory technique. It cannot be used that cannot be followed by farmers. Now, let us see what is a tissue culture and different types of tissue culture, and it is a completely laboratory technique until the individual plant is raised. It is called tissue culture. Now, let us see in a plant tissue culture. Plant tissue culture is nothing but micropropagation in vitro culture. What is a tissue culture? Tissue culture means it is a technique of growing cells, tissues, organs, all organisms in vitro and artificial culture medium under aseptic and controlled conditions. This is about tissue culture. Now, let us go to how tissue culture done in the laboratory. Before going to that, let us learn about what are the contributions for agriculture, for tissue culture. Let us see. He is an Aberland. He is the idea of the totipotency. He worked for totipotency in 1902. He is the Haberland. He was the first person of totipotency. And next, Murshige and Skoog. He was the first medium of the culture. And Maheshwari and Guga was the first Indian cultures. He found about embryo culture. Miller and Skoog, he was the first person of formation of root and like cytokinins and auxins. He was the first formation of root and shoot cultures. After that, White, he done the experiment with the root culture and he is succeed. Skoog prepared an artificial medium in 1962. This is about history, and let us see. Tissue culture had its origins at the beginning of the 20th century. At the beginning of the 20th century. At the beginning of the 20th century with the work of Gottlieb Haberland in plants. The first commercial use of plant clonal propagation was artificial media in the artificial medium in the and growth of orchid plants in 1920s. In 1950s and 1960s, there was a great deal of research but it was development of only for the tissue culture. Let us see. Now we discussed about what is the history of tissue culture. Now let us go for what are the requirements for the tissue culture. Tissue culture requirements are first one, appropriate tissue. Appropriate tissue is nothing but a tissue which is selected for desirable plant for culturing. And the second one is medium. Medium means a suitable medium containing energy sources and inorganic salts to supply growth needs, and it may be a semi-solid or liquid. Third one is aseptic conditions. The third requirement is aseptic conditions. Aseptic conditions are nothing but as microorganisms grow much more quickly at their culture and they can transfer and they can grow much more quickly in their culture. So, we have to need aseptic conditions. The fourth requirement is growth regulators. Growth regulators are nothing but cytokinins like auxins and cytokinins. The energy sources and salts are not sufficient for growing a complete individual plant. So, we need growth regulators. These are the requirements of tissue culture. Now, let us go deep into the tissue culture. The following steps are involved in tissue culture. Let us see. The tissue culture techniques: preparation of nutrient medium, sterilization of culture medium, preparation of an explant, inoculation of an explant, incubation for growth, and acclimatization of plantlets and transfer to pots. These are the steps involved in tissue culture experiments. Now, let us learn one by one. First, we go for preparation for medium. Medium. Medium is nothing but source for the growth. The medium consists of source, elements, other substances. Other substances source like carbohydrates, like sugars. Elements like mineral ions, like salts. And other substances like amino acids, proteins, vitamins, etc. The medium consists of source, elements and other substances. At source like carbohydrates like sugars, elements like mineral ions and like salts, other substances like any amino acids, proteins, vitamins, etc. These are all taken in a distilled water. This is called medium. These are all taken in a distilled water. These are all taken in a distilled water. This is called basal medium. Now, the basal medium consists of source, elements, other substances. This basal medium does not have any growth regulators. That is why it is called as basal medium. The mediums are different types. What are mediums of different types? Let us see. Liquid medium, semi-solid medium, and solid medium. Liquid medium means it sustains the growth of cell suspension. Semi-solid medium means if we convert the liquid medium is made into the semi-solid medium by adding some agar-agar. This is agar-agar. Adding some agar-agar is added to this liquid medium, it becomes thickened. Nearly the agar-agar is added 1 to 2%, and the solid medium means if we add more, that means 6 to 8% of semi-solid agar-agar to this, it becomes solid medium. These are the different types of medium. After that, the culture medium must be closed tightly with cotton plugs. The cotton plugs allowing the oxygen and carbon dioxide into test tubes. This is about medium. Now, let us go for sterilization. Sterilization is not. Here the culture medium is rich in nutrients, vitamins, amino acids, source, etc. So, we must be sterilize the medium with and the materials used, the medium and the equipment used for the tissue culture must be sterilized with the sterilizer called autoclave to kill the microorganisms. And it is carried out steam sterilizer called autoclave. See, observe here, it is our steam cookers are also used as an autoclave to remove the microorganisms. This is about autoclave. And we must be sterilize the mediums and equipments for 15 minutes at 121 degree centigrade and 15 pounds of pressure. Now, next go for explant. Suppose it is a plant. Explant is nothing but any part of a plant and that must be sterilized with detergents to clean the surface of the explant. Observe here, explant is nothing but any part of a plant that must be sterilized with detergents to clean the surface and washed with sodium hypochlorite to kill the microorganisms. Now, the explant is sterilized. Let us see. These are the sterilized explants, and we have just prepared a medium, sterilized nutrient medium. Now the explant is ready and the sterilized nutrient medium is also ready. Now I am inoculating the explant into the nutrient medium. This is called inoculation. See how I am inoculating the explant. Yes, and it must be put it in an incubator. This is an incubator. It maintains the temperature of 37 degrees Celsius, and I am putting the sterilized nutrient medium in an incubator. After 2 to 3 weeks, it becomes like a callus. Means the cells of explants, it becomes like a callus. That means the cells like explant grow and undergo repeated divisions, and undifferentiated mass of cells is called callus. This is called callus. Now I am taking another medium which has added with cytokinins and auxins to it, with high auxin and low cytokinins results in the formation of root formation. Observe here, the roots are formed. After few days, it becomes like a roots. This is called, the root formation is called rhizogenesis. The growth regulators with low auxins and high cytokinins results in the formation of shoot formation. This is called calogenesis. After that, the formation of calogenesis, rhizogenesis results in the formation of complete plant. See here, the plant is formed, a complete individual plant is formed. First we have taken a explant and then we can transfer it into a sterilized nutrient medium. Observe here, I am taking an explant and in a sterilized nutrient medium, I added some growth regulators to the medium and it forms like a roots and shoots and results in the formation of a individual plant. Sometimes the callus may directly develop into embryo. Embryo is used as future use and it can be directly used for culture. It is used also for culturing. This is called embryogenesis. Sometimes the callus may develop into the embryos. It is called embryogenesis. Embryogenesis are also called as embryoids, somatic embryoids. This is about tissue culture techniques. Now let us see in an History of plant tissue culture. The history of plant tissue culture first idea of totipotency was by Aberland in 1902. White established successful tomato in 1937. And Skoog and Miller demonstrated the role of auxins and cytokinins and root and shoot formation in tobacco. Murshige and Skoog introduced the medium culture in 1962. Guga and Maheshwari obtained the haploid plants through an anther culture. Different types of tissue cultures. Embryo culture, seed culture. Just now I am taken and a leaf and I am transferred a individual plant. Just like only we must be taken embryo or cell, etc. Callus, bud culture, organ culture, protoplast culture, meristem culture, etc. These are the parts of a plant. Tissue culture techniques. Preparation for nutrient medium, sterilization of culture medium, preparation of an explant, inoculation of an explant, incubation for growth, acclimatization of plantlets and transfer to plots. After this, acclimatization of plantlets. Acclimatization means the plant produced through tissue culture and transferred to fields. In a glassware, it is called acclimatization. After this, in the connection, we should also know about hybrid stomaconal hybrids. Stomaconal hybrids are nothing but fuse of protoplasm of different varieties of plants is called hybridization. Stomaconal hybridization. This is about tissue culture techniques. Let us once again repeat one by one. See how it is a plant and we have selected a explant. Explant is nothing but any part of a plant. Observe here. And medium. The medium is mixed with agar-agar. The liquid medium is nothing but cell suspension, and the solid medium is, if we add more agar-agar to it, it is called solid medium. Semi-solid medium means if we add less agar-agar to it, it is called liquid medium. In mediums we have supplied with source, salts and other substances like amino acids, proteins, etc., which is help for the primary growth of the cells up to callus. This is about medium. This causes the plant part to grow at a very rapid rates to produce new plantlets. It has been established the word chrysanthemum apex placed in tissue culture could produce up to 1 lakh new plantlets in 1 year. Different types of medium. Medium means nutrient medium. It is an autoclave. See, autoclave is used for sterilization at 121 degrees Celsius and 15 pounds of pressure. See how the explant is inoculated into the sterilized nutrient medium. See, it is also inoculating the explant into the sterilized nutrient medium. After that, it forms a group of cells is called callus. This is also callus. Undifferentiated mass of